just so there's plenty of time for questions at the end. And so
Sure.
Josh, Will is going to do the introductions and I'm going to mute myself.
Okay.
Okay, hey everybody. Just want to welcome y'all to the nutrition seminar webinar series. Sorry, tongue twister. I'm Will Florence and I'm a master's student in nutrition here at NC State. This week we'll be hearing from Dr. Josh Kellogg about unraveling botanical mixtures via untargeted metabolomics. But before we get started here, I just want to give a little background on Dr. Kellogg. Dr. Kellogg grew up in Los Angeles, California, and he went to the University of California, Berkeley, where he got his obtained his undergraduate degree in chemistry. He then went to University of Illinois for a master's degree in natural resources. This is where he met Dr. Marianne Lila. Dr. Kellogg worked under Dr. Lila at NC State, obtaining his PhD in nutrition. He completed his postdoc at UNCG in antibacterial work. He's currently an assistant professor at Penn State University and teaches both toxicology and metabolomics in the Department of Veterinary and Biomedical Science. Dr. Kellogg continues to work on natural products, chemical ecology, ethnobotany, metabolomics, data analysis protocols, and microbiomes. And just as a reminder, we'll have time for questions and answers at the end of the presentation. Just feel free to type them in the chat and we'll get to as many of them as we have time for. And now, without further ado, please join me in welcoming Dr. Josh Kellogg.
Thank you, everybody. It's good to be home, in a matter of speaking. I'm really excited. Thank you, Susie, for giving me the chance to come and share a little bit about what I've done since I left NC State. So this will be a mixture of some work that I started as a postdoc at UNCG in the lab of Najicek, as well as some of the projects that we've kind of commenced here at Penn State. Of course, we've had about a 9-month gap in the middle of that with our current pandemic. So, but things are moving forward, so we're excited about that. So without any further ado, let me share my screen. And there we go. Okay. There we go. Okay, so thank you again. I'm really excited to come home, so to speak, and share some of the work that I've been up to. And so yeah, and starting in my postdoc time, and it's evolved into my time here at Penn State, I've really worked on developing new metabolomics procedures and applying them to natural product discovery and chemical ecology. And so the question is, so metabolomics, for those of you who are not, who do not work in metabolomics on a daily basis, it is one of the newer omics kids on the block, so to speak. Most of us are very familiar with genomics, proteomics, transcriptomics, but metabolomics is really looking at the whole set of small molecules that are produced by an organism or a system. And one of the fascinating things, one of the things that really attracted me to this in the first place was that the metabolome is in constant flux, and it is a snapshot of all of the abiotic and biotic stresses and influence that an organism is experiencing at any given time. So in this example here with this tree, we see things like sunshine levels, precipitation levels, climate indicators, oxygen, carbon dioxide levels, predation from insects or herbivores that might elicit defense responses, parasitic infections by fungi or bacteria, or mutualistic interactions with fungi and bacteria, nutrient levels in the soil. All of this kind of comes together and causes the organism, any organism, not just a tree, this could be you as well. Hopefully you don't have any herbivores gnawing on you at the time, but I mean, who knows? But this really, the end result of all of this is that the organism produces this unique set of small molecules that is constantly changing based on how these inputs are changing. And so we can tell a lot about a system from its metabolome. Now, metabolomics in its theory is an unbiased analysis of all of the measurable small molecules that an organism is producing. As I mentioned, this is highly fluctuating because we live in a dynamic environment. All of us do, or any sample organism does. And so consequently, we get a lot of data out from our metabolomics experiments and studies. And I really find it fascinating, again, because it's a combination of analytical chemistry, botany, biology, biochemistry, and we do a lot of statistics as well. So any stats nerds out there, come join my lab. Now, even though the term metabolomics is only about 20-ish years old, 22 years old or so, the concept of how do small molecules predict a phenotype or predict changes in phenotype really go back centuries. And here are some, we see some early instances of the concept of metabolomics. The 13th Arab physician Ibn al-Nafis, his idea of metabolism was that the body are in this constant state of dissolution and nourishment. So they are always undergoing change. So this idea of flux in the metabolism and in the nutrient levels still holds true to this day. And then Ulrich Pinder, he produced this Epiphany Medicorum in 1506. And his work was looking at the colors, the viscosity, the taste, the smell, and yes, I did say taste, of urine, and how that relates to various disease conditions. And this is essentially the same thing that metabolomics is. We look at small molecules which impact color, which impact flavor, and we look at how does that relate to the overall phenotype, disease condition in this case. Luckily, thankfully, we no longer use the urine taste test as an analytical measure. So we're off the hook there, thankfully. And even though metabolomics is relatively new, as I said, the concept has, the codified concept has really been around for the last 20 years or so. It has found a home in many different disciplines, from drug discovery, disease pathogenesis, biomarker discovery in humans and plants and animals. Of course, precision medicine, precision nutrition are gaining ground, and metabolomics is finding a home there. Toxicology, drug studies, things like that. So, I mean, it's a wide variety of places where we found use for this, for metabolomics as a diagnostic tool. Now, when we're looking at metabolomics analyses, we really have kind of two approaches. One, the first being a targeted analysis, where we have a subset of this entire metabolome that we're going after. So this could be something like lipidomics, where we're looking at just the lipids that an organism is producing. It could be even more defined. We could be looking at just the leukotrienes that an organism is producing. And so the benefit of using this targeted analysis where we go after a subset of the molecules is that we're able to better quantify them. We know the structures of what we're looking for, so we can usually get standards. We can do more quantitative work. And then it can be a little more hypothesis driven. The flip side of that is untargeted metabolomics. We're venturing into the unknown, where we're trying to get the fingerprint or the overall chemical profile of the entire system, everything we can measure. And in an untargeted approach, we're not, at the outset, we're not as concerned about identifying every peak and every molecule in this metabolome. So we have a lot of unidentified features, but instead, we're using this profile to give us a global analysis, kind of a holistic look at the metabolome, and then do a lot of pattern recognition and comparative work between organisms, between control groups and wild groups and treatment groups. And this in the past has been more hypothesis generating, but the analytical tools that have been developed, especially in the last 6 years or so, have really let us not just generate hypotheses, but also answer a lot of hypotheses as well. And if we can use, this is not to say that these molecular structures and identifications are not important. We usually answer that as a secondary question. But at the outset, we can do a lot without intrinsically knowing the chemistry we're looking at, which can be important. And so in our lab, we do the vast majority of our work is untargeted. And I believe almost everything I'm presenting today is going to have an untargeted slant to it. And so why plants? I love plants, of course. I've been studying plants since I started in Dr. Lila's lab some number of years ago now. And one of the things that fascinates me about it at the time and still to this day is how complex plants are. So a human metabolome, is somewhere on the order of 3,000 to 5,000 small molecules coursing through your body at any given state, at any given time, and those are fairly conserved across people, across geography. Of course, we're all the same species, so that helps. But that makes identifying changes and identifying molecules much easier in a way, because we have such a relatively robust and stable set of molecules. Now, plants, on the other hand, an individual plant, estimates vary, and it could be anywhere from 7 to 15,000 small molecules in a plant at any given time. So already it's, almost an order of magnitude more complex in the plant itself. And then if you look at we obviously are looking at a lot of different species of plants. So the overall size of potential small molecules you could be looking at for a plant runs the order of anywhere from 200,000 to 1,000,000 different small molecule structures. And this is just plants, of course. And so, we have also such a wide structural diversity in the small metabolites and the small molecules that we have in plants, that I find just truly fascinating. So it's always tickled my fancy. So how can we use this approach now that you're all metabolomics experts? How can we use this tool to compare different complex systems? And that's one of the questions that we try to answer in our work. And so one of the case studies that I'm going to point out is kratom. I don't know how many folks are familiar with this plant, kratom. It is a plant from Southeast Asia, and some species also grow in Africa as well, Eastern Africa. But it is a plant that has gained some more attention in the last few years, and I'll say some notoriety in its use to, people have been using it anecdotally to try and wean themselves off of opioids. So people who have chronic pain, who have gotten hooked on opioids, they have been turning to kratom teas, kratom supplements, because they have several alkaloids present in them that activate some of the pain receptors, these new opioid and other opioid receptors in the body. But it's believed or hypothesized that they do not trigger the same sort of addictive pathways that fentanyl and oxycodone and all these synthetic opioids have. However, kratom falls under this kind of dietary supplement market, right? That's regulated by DSHEA, as I'm sure you've all talked about in your classes and learned about. So the regulation on kratom is fairly loose, and because of its potential analgesic properties, the FDA has been flirting with scheduling it. There's definitely groups out there that want to schedule it Schedule 1, which is of course the highest level of scheduling and restrictive access, saying there's essentially no medical benefit to the plant at all. And so they haven't done that yet, but every so often they'll kind of flirt with that idea. So there's not a ton of regulation in what's in kratom, commercial kratom products. And of course, they have the really interesting, fantastic names. There's red hippo Thai, and there's red vein Kali, and so it's very evocative of, shall we say, other medicinal plants, so to speak, which we do not study, by the way. So don't ask. But we were looking at kratom because it is purported to have these properties, but there's not a ton of literature out there about the chemistry of kratom, about the efficacy of kratom, and the authenticity of kratom. And that's where we really came in. So we were looking at commercial materials. We found 52 different commercial materials found on the internet, which was an interesting procurement process on its own. But we found 52, and when we profiled them, when we extracted them and we put them through a mass spec to get a untargeted chemical metabolomic profile of them, we found two real clusters. And so this is a PCA scores plot. And for those of you, again, who maybe are not in the metabolomics world and don't deal with these plots altogether, each dot represents one sample's profile. And so here we see K50, for example. And so we see all of the peaks, all of the small molecules that go into building, that go into their individual profile. And so whereas this K52, so we see some overlap, some peaks are the same. You know, we see this peak 399, peak here the same, but we definitely see differences. Like this peak here at 3 minutes, 3.15 minutes is noticeably absent in K50 right here. So the scores plot is a statistical representation of how similar these profiles are to one another. So the closer they are spatially in the plot, the more similar they are. The more further apart they are, the more different they are. And so we had two real groups that were manifesting in our commercial samples, which we didn't think there should be. It's all kratom. It all is supposed to be the same thing. But we definitely saw a difference. And you could see there were some differences in the chemical profiles. And so we started to look a little more into this and found that there were different alkaloids present in different types of material. So even though it was all being listed as kratom mitragyna speciosa, there were definitely chemical differences in there. And these were actually related alkaloids, which is what drives the bioactivity. So we partnered with a team actually at High Point University to do some binding capacity or binding affinity studies on the mu opioid, the kappa opioid, the delta opioid. And we saw that for some of the alkaloids in kratom, so mitragynine and the 7-hydroxymitragynine, they had noticeable binding, like measurable binding, and they were not too far away, especially the 7-hydroxy was not too far away, maybe an order of magnitude at most, away from the binding affinity of morphine, suggesting they do have analgesic properties, these alkaloids do. What's interesting though, is that the speciophylline, that extra alkaloid that was popping up in the K52 and those red cluster of samples has zero affinity for any of these receptors. So it has no analgesic properties whatsoever. And so depending on where the supplier is sourcing their material, you could be getting material that's very potent and is going to work for what you're doing. You're treating long-term chronic pain, or you're trying to wean yourself off opioids. Or you could be getting something that says that's kratom, it's labeled as the same species, but the chemical composition is completely different. And you wouldn't be seeing any effects whatsoever. So you could be taking as much of this as you want, which could lead into, we haven't looked at a lot of toxicology of this yet, but you'll be getting an effect. So this is the sort of, one of the areas where Metabolomics has been helping us in disseminating and describing source material for these kind of botanical dietary supplements. We see another example here, looking at actual adulteration. So not just, where we have a little more fine point to what we were looking at. So Hydrastis canadensis right here in the middle is golden seal. It's a fairly well-known antibacterial plant, grows in North America, grows in North Carolina, up in the mountains near Asheville. It grows up here in Pennsylvania too. And it's the root, the rhizome is actually what we use to grind up and make the antibacterial powder. It's an immune booster, anti-inflammatory. But of course, if you're harvesting the root, the rhizome, you're destroying the plant. And it's a slow grower. It takes about 4 or 5 years to get to a harvestable size. So there's been some evidence that other plants are being, spliced into golden seal supplements, such as this Coptis chinensis or golden thread. Grows much faster, less expensive, therefore, to harvest, has the same characteristic yellow rhizome, so it easily passes visual QC. And so this, I should say, is the work that was done by our then master's student, Diane Wallace. She has now gone on. She's the co-director of the mass spec facility at Chapel Hill. So she's actually just down the road from you all. So again, similar to the kratom, we took a bunch of commercial products looking at leaf material and root material, which should have noticeable chemical differences, but we still wanted to profile both types of samples, as well as verified reference materials, 2 of which we had a friend who grew them, so we knew, we harvested known material, and some were verified commercial materials. And so again, looking at this scores plot, again, the more distant points are away from each other, the more different they are. We saw 3 samples that really fell outside our 95% confidence interval for our data, which really struck us as strange, again, because it says golden seal on the label. We expect it all to be, relatively similar. And for reference, the dark green are our reference material, leaf material, and the dark blue are our root reference material, rhizome reference material. So again, using an untargeted approach, we can then go in and look a little more at the chemistry and see that the samples that were being pulled further away had high levels of these other sort of alkaloids, coptacine, palmitine, dihydrocoptacine, which are definitely not present in golden seal. So this, of course, triggers your warning, your panic button. And they were noticeably low in cannadine and hydrosine, 2 of the more well-known alkaloids that are present and specific to golden seal. So we actually went through and did a quantitative analysis. We did a targeted metabolomics analysis as well, where we quantified all of these 7 alkaloids and all of the plant material, and we added in extra reference materials to try and tease out what was going on. And we found that this GS33 that is clearly not golden seal actually lines up really well with Coptis chinensis. So it is completely adulterated. The GS7 also was a mixture of Coptis chinensis and another plant altogether, Mahonia aquifolium, or Oregon grape. And then GS20 we found to be a blend. So it had golden seal. It also had some of this Coptis chinensis in there as well. So this is a great study, and it was really interesting for us to be able to show that this kind of, that is unfortunate, of course, but it was interesting at the same time to see that an untargeted metabolomics approach could pull out these outliers from the data set. But it led us to the problem, or to the question is, we went in knowing we knew a lot about golden seal. It's a fairly well-studied plant. We kind of knew the plant that we thought might be the adulterant. And so we had a lot of foreground knowledge. And so the question is, of course, what if you don't know what you don't know? We do a lot of looking at plants that have very little background into them. Even things like kratom, which are much less studied than golden seal. And what we found is, one of the problems that we saw is using this PCA scores plot to drive our conclusions, or our next hypotheses. When you run a PCA model, it actually creates anywhere between 5 and 50 components of the model to represent all of the data that you're looking at, but you can't make a 50-dimensional graph, and where that's not possible. So everyone who does metabolomics condenses this down into 2 dimensions. And the selection of these 2 dimensions we found looking at other papers is really an ad hoc kind of process. You choose the 2 that makes your data look the best. Well, that's awesome, but as a scientist and as an analytical chemist, and it doesn't sit right? You're like, how can you just pick 2 and that's what you're going off of? And when we looked at our golden seal data, we saw this. So, I said we combined, we had leaf material and root material. And as a plant biologist, you would expect that those chemistries should be different. And so if you didn't know about the adulteration, if you're going into this blind, you would probably pick a scores plot where the leaf and the root material were fairly well separated, like this one right here. But what you see is actually our outliers. 2 of them get swallowed up into those plots now. You would never pick them out as outliers. You would look at that and be like, okay, so this GS20 is kind of weird, and that's it. And so it seems like no matter which pairwise comparison of principal components we choose, in a way you can, not intentionally, but you can manipulate what you're seeing. And through, you could be completely innocent of this, but you could be completely biasing what you are actually putting out there as your data. And so what we went ahead and did is we try to combine these multiple components into a single metric, which we call the composite score. So first we had to figure out how many of these model components were actually significant in driving the model creation. For the golden seal, it was 5. And then we created a metric where we integrated all of the those 5 PCA components into a single correlative metric. And here we see a pairwise comparison across every sample in our data set. So every sample is compared with every other sample, and it's given a correlation score of -1 to 1. Of course, 1 being perfectly aligned, -1 being perfectly anti-aligned. And that we liked it. It was good. It gave us numbers. That's cool. We all like numbers and data. But we also went back to check to make sure that this actually tied back to the actual chemical data, because our fear was we were doing so much data manipulation that it had no relevance to what we were actually measuring in the mass spec or in the sample. But what we found is when we looked at samples that had a very high composite score, the spectral overlap from the chromatograms of the mass spec looked really good. We see a lot of the same peaks, same shapes, same intensities. And when we saw 2 samples that had a very poor correlation score or composite score, we see a distinct shift in the peaks. Their distribution, their overlap is very poor. So this gave us good confidence that what we were measuring and what we were reporting out actually had some kind of real life relevance to what we were looking at in the first place. Of course, looking at this is great for comparing two samples against each other, but to do an overall analysis, I mean, one of the beauties of PCA is that it is so clean graphic. And so we actually took this composite score and turned it into a network analysis graph. And what we see here is we see the root material here in yellow with the standard right here in the center, and they're all connected. We see the leaf material, which has some connections because there was some overlap in chemistry, which we'd expect, but they are distinct in their own cluster with the reference material kind of centrally located. And then we see our 3 outliers definitely pulled away. The 2 that had no golden seal in them, GS33 and GS07, have no connections into that network graph. Whereas GS20, which was a blend, had some connections in because there was some golden seal. And so we believe this is a really interesting tool that we keep using and we're continuing to use to do this kind of comparative. So those are some examples of how we do comparative metabolomics and how we use untargeted approaches to compare botanical systems in a way. But inevitably, no matter what sort of study we're doing or who approaches us, the question always is, well, what molecule is doing this? Like, who's responsible? How do we know what is the chemical underpinnings of whatever phenotype change we're seeing, bioactivity we're seeing, what have you? So a lot of our work also goes into that. And this is important. I mean, when we're looking at a natural product or you're looking at a complex sample, we often have very little amount. And so if you see a profile like this on the screen and you have a bioactivity that's antibacterial, it's anti-cancer, it's antioxidant, the question naturally, hopefully, arises is which of these peaks is responsible for the bioactivity that we observed? And it'd be great if we could isolate every peak and test every peak and study every peak, but let's be honest, nobody has that kind of time and resources. So we have to choose. Now, the way that it's done in the past is this gold standard for drug discovery from natural products is an iterative process where we take a plant extract, we make fractions, we test the fractions in our bioassay, read the results, and that tells us, okay, fraction 4 is the active fraction. But it's still not a pure molecule. It's still a complex mixture, just less complex. So we go through this process again. We make subfractions of the fraction and subfractions of the subfractions, eventually until we get to the point where the active is 1 molecule. Oh. And this, again, has been the standard for decades. I mean, all of our well-known anti-malarial drugs, anti-cancer drugs, all of them, I would say virtually all of them have come from this sort of approach. The problem is, though, it is incredibly resource heavy. It takes a lot of resource, a lot of time, a lot of energy. And we have many instances where you don't get to 1 molecule. You go all the way down and it's not a molecule. You've either lost it on the columns, it's broken down because you've gone through this process so many times. Maybe there was synergy. Maybe there were 2 molecules working together. Now you've separated them and you lose that activity. So there's a lot of caveats to this approach. And that is one of the reasons why some of the drug manufacturers about 20 or so years ago started pulling back from natural products. I thought it was too resource intensive. Wrong. So what we've done instead is we have used our untargeted metabolomics that we can do this profiling, and we combine it with the biological activity and use some multivariate modeling and some machine learning techniques to really drive identification of potential bioactive molecules. And so we developed in conjunction with our collaborator, Olav Kvalheim, who's at the University of Bergen over in Norway. And so the biochemometric modeling that we do, we use the bioactivity to drive the clustering and the separation. So it's very similar in a way to our PCA analysis that we saw earlier, except we're using a lot of the bioactivity to help separate active from inactive in conjunction with the chemistry that's going on. And so we're hoping, then we have metrics that take that model and pull out which of the features, which of the ions or metabolites maximize their covariance and correlation with the bioassay data that we're putting in. And so here I have an example. So we were looking at antibacterial compounds from fungal endophytes from golden seal of all things. Can't get away from it. And so we grew up several different fungal endophytes, and our initial extract was really high in inhibiting Staphylococcus aureus. We did a first round of fractionation, fraction 2, again, complete annihilation of the bacteria. Awesome, we were very excited. I should say this was done with our undergrad, at the time our undergrad, Sochima, who is now actually at Wake Forest Med School. So people off doing cool stuff. We took fraction 2, subfractionated it. So again, we're seeing this iterative process. We saw subfractions 9 and 10 still had really high activity, but when we profiled them, we were still not at a single molecule. They were still about a couple dozen or so peaks that we could see in the mass spec. And we were getting down to the end of our material. Endophytes don't grow particularly quickly. And so, we were at sub-milligram levels of material that we have left. So we put this into our biochemometric analysis. We put all of the fraction data with the bioassay and the mass spec, combined it into this partial least squared multivariate modeling, which we could see did a pretty good job at pulling out the active extract and fractions against the inactive ones. But looking at some of the metrics that come out, this S-plot predicted 2 main bioactive molecules, a mass of 273 and a mass of 400, whereas the selectivity ratio analysis pulled out just one, the 400 peak. The 422 peak that we see here is the sodium adduct, so it's not a real peak. And this 400 peak is alter-cetin. It's a derivative of the equicetin. It has been discovered, but there's not a lot of work done. And so we actually went and looked at the activity of the 273 peak. This was alternarial, as well as our 400 peak, the alter-cetin. And we saw that against a susceptible staph, the alternarial had very blah sort of activity, about 275 micromolar, nothing really to write home about if you're looking for antibacterials. Alter-cetin, on the other hand, had sub-micromolar activity against the susceptible staph, and against a resistant staph, against MRSA, the alternarial had 0 activity, whereas alter-cetin had low micromolar activity. So something interesting to look at. But I think what really sold it for us is if you look at the chromatogram of the original extract, and I asked any of you to pick which peak is the alternarial and which peak, most importantly, is the alter-cetin peak. You'd have a range of guesses, and I promise you, I would not guess this one either. But the alter-cetin peak, there's our alternarial, the one that had this kind of ho-hum activity. That's the alter-cetin peak. And again, if you were going through the iterative bioassay guided fractionation kind of procedure, you might never get so far as to isolate this one on its own in any appreciable quantity. So I think this really spoke to the ability of our biochemometrics approach to enhance our selection of priority candidates, and really increasing the rationale behind why we're going after certain compounds. So we can do targeted isolation And then we can go through structural elucidation if we need to, or in bioactivity studies. So we've since applied this to other systems as well. We were looking at green tea and how it can inhibit phase 2 metabolism in the gut. There's a big question of how dietary supplements, especially botanicals, could influence drug metabolism. And the fairly well-known case of grapefruit inhibiting the CYP450 and interfering with warfarin metabolism and things like that. So again, we could use the biochemometrics similar to approach by fractionating or making simple fractions of the green tea and profiling them against glucuronidase activity. Oh, we got too many arrows. And we see our biochemometrics pulled out six peaks from the green tea. Luckily for us, they were all adducts and isotope peaks of one particular catechin, the epicatechin gallate. And we did follow-up studies with all of the main catechins found in green tea, and epicatechin gallate had the highest binding affinity with the glucuronidase enzyme. So this helped kind of confirm our predictions from the biochemometrics that we were finding the most bioactive principle, what was responsible for green tea's interference with the phase 2 metabolism. So moving to new projects, some of the stuff we're embarking on is looking actually for new ligands in the diet and from the gut microbiome for the aryl hydrocarbon receptors. So this AHR is really important to maintain gut integrity and gut homeostasis. It's also a big one in protecting the GI tract, especially the intestines, from toxic exposure. And work that has been done has found that the AHR responds to a variety of different ligands, some from the microbiome, some from the diet, and of course some endogenous compounds as well. So the question is, how can we again be more effective in our screening process, in our discovery process? So the way that we're doing this study is we're actually looking at fragmentation patterns to identify similar molecules, to look for structurally similar compounds to what we know activate the AHR. So we're using a known ligands to kind of seed a mass spec fragmentation network. And then we can plug in our untargeted metabolomics data into this network and look for compounds that might be the same. So we find we're dereplicating, we're finding what's already known, as well as finding structural analogs that could be novel, that still could be AHR ligands. So as kind of a proof of concept, we actually used a study from someone else in our department, Andrew Patterson, who looked at white button mushrooms and their chemistry of white button mushrooms that activate this AHR. And so we used his raw data blinded. We were blinded, and we used his raw data, combining it with our preliminary seeded network, and came up with a molecular network. So this network, any points that are connected, share some structural similarity based on their mass spec fragmentation. And if we look at this subnetwork here, this little tiny guy right here, and blow it up, we color coded it so that compounds that are red are from our peaks that are found in our target, our library, so to speak, our seeded network. Compounds that are blue are found from the mushroom untargeted mass spec analysis. And then purple, of course, red plus blue is purple. So these are ones that have overlap. They are compounds that we seeded into our network that are also found in the mushroom. And so what's interesting is some of these benzothiazole derivatives, this red one, this hydroxy one, was not found in the mushrooms, but we know it has activity. These two, the thiol and the amino derivatives, were found in the mushrooms. And what's really interesting is we found this peak here that has activity. It should be structurally similar to this benzothiazole structure, but we don't know what it is. So we're following up on this. And so other work that we're doing is looking for the next generation of bioactive molecules. And so where do they come from? Of course, I'm a natural products person. We've looked to nature for so long for diet and health and medicine. So I'm kind of maybe a little biased, but I think the numbers prove us out. If you look at all of the small molecule drugs that are out there currently, about almost 2/3 have their roots in nature. They are direct natural products. They're natural product analogs, semi-synthetic derivatives, but they have their roots of their discovery in nature. This bumps up to 75%, 3/4 if we're looking at small molecule chemotherapy or antibacterial compounds. So, there's a lot that we've been able to mine from the natural world to help promote human health. And this, of course, is just on the pharmaceutical end of the side. And the Kew Botanical Gardens about 3 years ago or so, they did a survey and found that there's about 28,000 plant species across 90 countries that are used as medicine, but less than 10% of those have really been looked at for their active chemistry. So there's, in the natural products world, we face, there's this, everyone loves to go after new environments. So there's a lot of, people love marine natural products and extremophile natural products and bacterial natural products. But I think this highlights the potential that plants still have to yield new, interesting chemistry that could be, that could help maintain health. And even looking at North American plants, of course, we think of natural products, we think of medicinal plants, we think of, Sean Connery in the Amazon with his GC, roughing it to find the tropical plants. But, looking at plants that are found here in North America, there's still a large reservoir of plants that have not really been rigorously looked at or reported on. So we're looking at several plants right now found in the southwest of the country. We got some help from the UC Berkeley Botanical Garden in procuring samples of these to do some preliminary studies, which we're excited about because they obviously don't grow in Pennsylvania. And we are also embarking on other plants that have well-known activity, but have potentially new use. So one project that we're working on with the Worcester Polytechnic and Dr. Scarlett Shell is looking at Artemisia. So Artemisia is famous for the artemisinin, the anti-malarial drug. But she doesn't work on malaria, she works on tuberculosis. And the preliminary data shows that the artemisinin is decent at reducing the infection of tuberculosis, so it reduces colony density. But what we see is the plant extract actually works orders of magnitude better than just artemisinin. So this suggests that there's something else in the plant that is either synergizing with artemisinin to improve its activity, or there's another chemistry altogether going on. So we just were awarded an R21 from the NIH to look at this system. And we're also looking at how Artemisia could play into other diseases as well, very timely diseases like COVID-19. So looking at a variety of cultivars of Artemisia against SARS-CoV-2. In lung cells, we see that there are various levels of efficacy. But what was interesting in this study especially is that it actually looks to have nothing to do with the artemisinin, because as the artemisinin concentration increased, the IC50 or IC90 increased as well, meaning it got less efficacious in preventing the invasion of COVID into this lung cell in vitro culture. So again, that suggests that there's something else in the plant that is efficacious. And so this is currently under review at the NIH as well. And so for this, we're looking at using our biochemometrics, but we're also using a comparative bioactivity model. So mapping the concentration of artemisinin and then using that to deduce how much activity is actually derived from the artemisinin concentration and how much is due to other chemistry found in the plant. Looking at plants, not just in North America, but we are actively looking at, we have partnerships with several different groups, and we're pursuing groups in a couple different areas of the world. We have collaborations that we're working on with Madagascar, Peru, Ecuador, Belize, as well as Native American groups here in the United States, which we're excited to still be pursuing. I'd say the last area that we're starting to look at in terms of developing new potential efficacious treatments is the microbiome. So we are all very well familiar, I imagine you are well familiar with the gut microbiome in humans, but of course plants have a microbiome as well. They have an endophytic microbiome that's inside the plant. They have the rhizosphere and soil microbiome. But we're looking at one of the reasons why we're so interested in this is because as natural product discovery has matured, we found that what we thought the plant was producing, we saw some very famous drugs and bioactive molecules that were assumed at the beginning to come from plants. The most famous, of course, and with a very strong North Carolina connection, is Taxol, the drug from the Pacific yew tree, which was discovered at RTI just down the road back in the 70s. Very famous anti-cancer drug, saved who knows how many millions of lives. But what they've found since then is actually due to fungal endophytes that grow inside the tree. Now, in the 70s, they didn't know this. They ground up the tree tissue and they found the drug. But they've since found that the tree doesn't have the biosynthetic mechanisms capable of producing Taxol, but these fungal endophytes do. So these microbiomes that are in and around the plant have the potential that could be actually the ones doing a lot of this biosynthetic work, this natural product synthesis that we're so interested in. My arrows. Again, St. John's wort. Again, a lot of the hypericin and some of the antidepressive activity that we attribute to St. John's wort also is believed to come from several endophytes found within the plant itself. So we are looking at, I have so many slides, I have so much to say, I can't. The benefit is we've found in previous studies that a grad student in our postdoc lab did back in 2016 was that even though the concentration of endophytes, the concentration of this microbiome is very tiny, we are able to see their metabolites produced from a macro level. So grinding up a plant, there is the potential to see, quote unquote, to measure levels of these bacterial or fungal produced compounds in the plant tissue, which means that if we can see them, then they have the potential to actually have efficacy at a real level. So it's not like we have to, what we could be attributing to the plant as having a certain bioactivity could in fact be coming from the microbiome, from the microbial community. And so with that, we're looking at the phylosphere, so the leaf microbiome of certain medicinal plants. Creosote or Laurea tridentata is our first. And so we're looking at, does the microbiome on the leaf surface, of course, they most likely produce bioactive molecules as part of their innate competition with one another for resources from a chemical ecology perspective. But do they produce anything that is relevant to human bacterial conditions? That's the question that we have here. So looking at we had a Laurea tridentata where they were sterilely washed. And we compared the leaves with the microbiome intact and the leaves without the microbiome. And we saw, at least our collaborator, Dr. Brittany Kruger, who's at the Desert Research Institute, they ran the preliminary bacterial studies. We did see the variant with the microbiome had a larger clearance of the bacteria from the plate than without the microbiome. So again, suggesting there's something in the microbiome, the leaf microbiome, that is bioactive. We took those samples and started comparing them from an untargeted metabolomics perspective. And what we saw here is we definitely saw peaks in the unwashed, so with the microbiome intact, that disappeared after the washing was complete. So suggesting that there is chemistry that the microbiome is producing that is lost, and that could be responsible for the bioactivity. This project is ongoing. Whew, that was a lot. Sorry, I got talking and I just can't seem to slow down. So, kind of wrapping up, I hope that this kind of showed how metabolomics can be a very powerful analytical tool to investigate complex chemical and biological systems. And so some of the work that we're embarking on is also improving metabolomics itself, something like the composite score that we worked on earlier, ways that we can do better data processing, feature annotation, as well as cross-platform integration, merging the benefits of mass spec with NMR or different chromatography systems. Hopefully some of these studies showed the glimpse of how combining our metabolomic profiling with biological data really gives us a great data set that we can mine for potential bioactive structures. And then hopefully I sold you on the fact that plants and their associated microbiomes, of course, hold a lot of potential for developing new chemistries and new therapies to combat chronic as well as infectious diseases. So with that, I'd love to thank a whole lot of people. Of course, my nascent lab at Penn State, we're so new, we don't even have a picture yet. But I'm really grateful for them for sticking with me through this pandemic and taking charge for a lot of these new projects that we're embarking on, which is awesome. My postdoc lab in Greensboro, the most recent incarnation of us who set me on this, after NC State set me on this path. And then the initial funding I've received from the American Society of Pharmacognosy and the NIH, some of the work that you've seen here, and our collaborators at the DRI and the WPI, and the UC Berkeley Botanical Gardens and the University of Washington, and the list, of course, goes on and on. But thank you for your attention. I hope that was not too whirlwind of a, of a talk, but I would be really excited to answer any questions that you might have.
Thank you so much, Dr. Kellogg. That was fantastic. Now we're going to do a brief Q&A session, and we'll get to as many questions as we have time for. So if you have not already done so, please just type your questions in the chat, and we will go with the order that they come in. So let me pull up the chat. Okay. So first we have Theo. I find it mind-boggling how many unknown metabolites show up in blood, intestines of our animals. Okay, shows that there is still a lot up to be discovered. Thank you, Theo. And
it is very true. It is, I study plants, but I have other faculty here who do metabolomics on animals and humans, and it is I said there's only 3,000 to 5,000 metabolites, but that is still by no means trivial in any way. So they have a lot of work cut out for them as well, for sure.
Dr. Goodell says it's clear that there's a lot of stats knowledge is required for these analyses. Are you running these stats yourself, or are you partnering with a statistician? She said, if it's all you, what training did you have to do to be prepared for this work?
That is a fantastic question, because I am not a statistician by training in any shape, way, or form. So we do run the analyses ourselves. So I've done a lot of self-teaching, we code most of this into R, and so we run most of our stats in R nowadays, which is fantastic in a way because it's open platform, and there's a lot of other smarter stats people out there who have built packages that we can use. Some of the statistics that we develop, like the composite score, we work hand in hand with a statistician. So Olav Kavalheim, our Norwegian chemometrician, has been really integral in making sure that the stats and the math that we're proposing and the things we're dreaming up actually work and come to fruition. So he's been a fantastic collaborator. But the modeling that you see here, the PCA modeling, the PLS modeling, the selectivity ratio, all that work we do ourselves. And so, as I said, I was mostly self-taught. I'm encouraging my students coming into my lab. They are taking more statistics courses than I did as a graduate student. And we do not need to be statistics wizards necessarily, but I want them to have an understanding of what's going into the models that we're making. So again, for me, that came from reading dozens and dozens of papers and getting textbooks from the library and such like that. But I'm moving it more into coursework for them.
Awesome. Next question. In addition to the stats question, if they're just picking the best graph that represents the data, how can we trust that their conclusions are valid, and how widely accepted is the new composite score method becoming?
Excellent questions. And that's the thing is, before we ran into this golden seal problem, we hadn't, I mean, you read a paper and you just, I, again, not being a statistician, I didn't know this at the time. I just accepted like the PCA is a PCA and that's what it looks like, and ta-da. And it didn't occur to me until then that the choice of model components, how drastic the choice of model components has on your overall data presentation, as well as how much of the data you're representing. Of course, every component models a certain, theoretically models a certain percentage of the data. And so you could construct PCA graphs that look fantastic, but they're only modeling a small percentage of the actual variance in data that you're trying to capture. And so how do we trust them? I think there's a couple ways you can think about this. I mean, the good thing about Metabolomics is we're doing a better job now at releasing our data to be publicly analyzed. So most of our data sets are out in public databases, so someone could check our work, and that's well and good. We don't always have the time to check every paper that we read, so you have to hope that they're following best practices. As far as the composite score, we just published that last year, so it's still in its infancy, I will say. Hopefully people find it to be a worthwhile thing to look at. I was chatting with someone who's looking at to use it for the wine metabolomics. So it might be catching on. It's hard, I think it's too early to say. But again, that code was done in R and it's all freely available up online. So, yeah.
Awesome. Next question. You mentioned collaborations with Ecuador and Peru and South America. Have you considered the Central America region? Maybe there are some different plant species to be considered.
Honestly, if I could have my dream job and not have to worry about funding, I would just form these collaborations and kind of trot all over the place and look at cool new plants. Unfortunately, that's hard to get funded. But we do have, we're trying to get some partnerships started actually in the Belize area. There are several faculty here at Penn State who work in Belize doing plants and agricultural techniques. And so we're looking at how we can partner with them to piggyback and try to form some partnerships. I know that, of course, Jillian de Gazelle, who's at NC State, has a long history looking at Belizean plants, cacao, and things like that from the Mayan area. And so she and I always are in communication about partnering up as well. So hopefully that will come to fruition. But yeah, there's definitely different plants for sure. And all plants are new plants, and all new plants are exciting.
Great. We have a couple more questions. Do you have a few more minutes,
Yeah.
Dr. Kellogg?
Yeah,
Okay.
I know I'm running. I'm talking too much. I'm sorry.
And just to be respectful of anyone's time, if you have to leave, totally understand. So I got two more in the chat here.
Sure.
And thank you so much, Dr. Kellogg. It's interesting how metabolomics can unpack insight into the bioactivity of different compounds. I'm interested also in understanding how the analysis of compounds can indicate upstream changes in plant and animal health. How useful is our current understanding of metabolomics in predicting health and function of the overall system? For example, plant, soil, animal gut, etc.
That is a great question. So if I'm understanding this, if you're trying to relate changes in metabolomics back to overall animal health or human health. There's a couple ways that can be done. There is a lot of work that goes into biomarker identification for certain disease states, whether it's infectious disease or cancer biomarkers or things like that, that can give us an indication of the overall health in respect to a particular disease. Thinking more broadly, we can also look at using the kind of untargeted metabolomics to, especially in controlled studies and controlled environments, we can use this data to look at what we would consider healthy specimens versus unhealthy, and look again at pattern recognition to see what's changing, even if we don't know the chemistry. Of course, we'd love to know the chemistry. So I think it is definitely possible to use this, and as I mentioned very quickly at the beginning, that metabolomics is finding a home in things like precision medicine, precision nutrition. We have a project that's under consideration for funding using metabolomics to do dietary intake studies. So, food recall studies have, of course, their caveats and their limitations, but could we use metabolites from the food, and could we kind of back calculate? Yes, I saw that you had a chicken burrito 2 days ago. Ha ha ha. So there's a lot of ways I think that metabolomics could be used to describe health in a variety of ways.
Thank you. And last but not least, you showed us that some of the products on the market may not actually contain the material it says, such as with the golden seal. Is this something the FDA should be in charge of checking? And are there too many differences in those metabolites to fully know what you're looking at in those products?
Good questions. So as far as the FDA's, this falls into a very murky gray area as far as regulation of dietary supplements and botanicals in general. Currently the FDA is really only responsible to investigate if there is a credible health concern. So we think of like ephedra back in the 90s, early 2000s kind of time frame, when this dietary, it was an exercise supplement, shed weight really quickly, but they found that it was stressing the heart. And so they had, it was an epidemiological study really, it was kind of tying heart attacks and morbidity back to taking this ephedra supplement. But really their only focus is on safety, not efficacy. So if you take a Golden Seal product and it's not as anti-inflammatory as real Golden Seal, as the laws, as my understanding, I'm not a legal or policy expert, but my understanding is the way that DSHEA is set up right now, that's not the FDA is not really set up to regulate that. Is it unfortunate? Yes. Could it be changed? Yes. Should it be changed? That's a long topic that we'll have to save to another time. As far as too many differences in the metabolites, I think that, especially if we're looking at known botanicals or known dietary supplements, there is enough known of the chemistry to say, like for the Golden Seal, we can say definitively the sample should not have, coptacine or palmitine in them. And so if we're finding those metabolites in a mass spec, and an extraction in a mass spec profile, we can definitively say like there's something in there that is not Golden Seal. Now you could, if you could go further and quantify them and figure out, okay, this is 20% palmitine, there's a 20% adulteration or 50% adulteration. But I think we do have enough analytical power on the metabolomic side as well as through follow-up studies to say definitively, yes, there is definitely adulteration going on.
And one last follow-up question to that is, do those metabolites vary region by region?
It depends on the plant. It'll depend on the system. Oftentimes what we see, they don't necessarily vary in terms of their structure. Like, Golden Seal is not going to suddenly start synthesizing coptacine if we grow it in California versus North Carolina. You'll see relative changes in the compounds that it's already making. And yes, it definitely varies region by region. I mean, we did a green tea study, and I did a lot of reading about green tea. And I mean, The chemical composition of the tea varies by climate, by geography, by elevation where it's grown, by the time of day that it's picked. Is it grown in the shade or is it grown in the sun? I mean, like, there's so much that goes into it. The chemistry is, so dependent on every little biotic and abiotic factor. But yeah, it definitely can vary. But again, like, what's coded in the genetics is not going to change. So the biosynthetic mechanisms are not going to spontaneously change to make something that we haven't seen before. I don't think.
Thank you. I got another one in here. If you're okay with that.
Mhm.
If you don't think it would be appropriate for the FDA to regulate dietary supplements and botanicals, how would you suggest we address issues related to quality control of products and efficacy, especially with more and more people choosing natural products?
Well, I didn't say it wouldn't be appropriate. I said that right now the structure is not set up for them to do that. That's not their legislative directive is not to follow a lot of these issues. Is it appropriate? I think it is, because from a consumer safety standpoint, we definitely, we do need to have knowledge if there are adulterations going into our botanical dietary supplements or dietary supplements in general. And yes, I mean, more and more people are turning to these, alternative remedies, shall we say, or natural products to maintain their health. There are growing groups who are doing this kind of work, and they don't necessarily have legislative or regulatory power to do this, but they are still putting out information to inform consumers. There's several groups in the now I can't remember the acronym, but they're environmental health folks who are doing toxicological work on dietary supplements and working on drug interactions. There's a whole office of dietary supplements at the NIH that's looking at safety and efficacy of these groups. And so there is definitely a need to address adulteration and supply chain issues and things like that. It's just currently not the purview of the FDA. And that, of course, can change, especially if we, the people, decide that we want those priorities to change. And yes, it's definitely appropriate that somebody should be keeping tabs on this, because it is such a growing market. It's a difficult question, though, and again, it's because it's how the DSHEA is set up is what makes it so difficult currently.
Great. Thank you so much, Dr. Kellogg. So next week we will have, at the same time and place, we'll have a presentation on precision nutrition strategies in poultry by Ben Harmon. And if we could, before we adjourn, please join me in another round of applause and thank you for Dr. Kellogg.
No, thank you all.
Thank you so much.
Thank you.
Thank you, guys. It was such a treat to be able to come back and chat and see all of your wonderful faces again. Well, a lot of new faces, but a lot of wonderful familiar faces as well.
We're so glad you could come join us.
Thank you.
It was good to